Hello, and welcome to this webinar on the role of INKmune in the arming of NK cells to target solid tumours in the tumour microenvironment. I'm Mark Lowdell. I'm the CSO of INmune Bio and the developer behind INKmune. As you know, INmune Bio is a publicly listed company, so I encourage you to read our forward-looking statement before proceeding with this webinar. So I've been interested in what happens when an NK cell interacts with a tumour cell for many years, and here are some publications from my academic group before the formation of INmune Bio. And what you can see on the left-hand side here, the Journal of Immunology paper from 2007, when we incubate NK cells, resting NK cells from healthy donors with the CTV1 tumour cell line, and I remind you that CTV1 is the parent cell line from which we generated INB16. INB16 is the cell line owned by INmune Bio. And when we make it replication incompetent, we term that INKmune, which is the drug that is in clinical trial. So you will hear me use the three interchangeably, I'm afraid, throughout the talk, but bear with me. So as you can see here on the left-hand side, the CD69 plot, when we incubate NK cells with CTV1 here, we get activation of the NK cells within an hour. It peaks at 3 hours, it's sustained for 6 hours, and indeed beyond that. But more importantly, the plot next to that shows the ability of these NK cells to kill the NK-resistant tumour cell line, Raji. So NK cells incubated with CTV1, the far side of that plot, this is 20 or 30 donors here, you can see a substantial increase in the ability of those NK cells to kill the NK-resistant Raji cell compared to the resting NK cells on the far left-hand side, labeled nil. And this is actually specific to CTV1 activation. If you use two other different tumour cell lines, U937 and HL60, these do not prime the NK cells to kill Raji cells. So there is something that's relatively unique about CTV1 in doing that. The plot directly beneath that shows the importance of this in a solid tumour setting. And here we've got NK cells from 5 or 6 ladies with ovarian cancer, and showing that they have very poor ability to kill the ovarian cancer cells. But if we prime them with CTV1 overnight, we get substantial increase. And the same is true, as you can see, of patients with breast cancer. We show very poor killing of breast cancer cells in the absence of priming. But once they've been primed with INKmune, we get substantial increase in that killing. So the first evidence way back in 2007 that INKmune actually primes NK cells to kill solid tumours as well as liquid tumours. The next paper in 2011 on the right-hand side there really is the first paper showing our beginning to understand the mechanism of action of INKmune. And this becomes critical to us filing this ultimately as a licensed medicine. So to have these data from early on in its development is very important. And I'll just point you to the bottom two graphs on that plot. If we look at the bottom left-hand one, it starts off looking at the ability of NK cells to kill Raji cells. And you can see in that black bar, it's very poor. But if we make the Raji cells express a molecule called CD15, which they don't normally express, you can see you can overcome that inhibition of the Raji cells, and these Raji cells now are killed by resting NK cells. They get the additional signal that they need. And indeed, if you block that CD15 with an antibody, you ablate the killing. So this demonstrates to us for the first time that CD15 is critical as a co-stimulatory molecule on the tumour cell in terms of priming them for NK killing or making them susceptible to NK killing. So what we've then observed is that when the NK cell binds to INB16 or CTV1 or INKmune, it actually transfers the CD15 molecule from the priming cell, and it carries that molecule with it. And you can see here in that plot that resting NK cells don't express CD15, but after they've been incubated with the CTV1 or INKmune, they do express CD15, about 15% of them. Interestingly, a similar percentage also picked up PKH, and that's the experiment that you can see in the plot in the photograph on the right-hand side. Here you have a tumor-primed NK cell generated by interacting with INB16, but the INB16 membrane was previously dyed with a green dye called PKH. And what you've seen there is that upon activation and interaction with the INB16, elements of the membrane of INB16 have transferred into the membrane of the NK cell during that priming event. And so the NK cell goes away carrying some of the proteins from the INB16. The technical term for it is called trogocytosis. It's been observed by other groups, not just ours. But we believe this is arming the NK cell, not only so that it can now kill NK-resistant tumor, but it carries those proteins to interact with other resting NK cells and expand that interaction. And I think that is what we're seeing in the next slide. So here we've been looking at 2 clinical trials that were done before the formation of INmune Bio, with what we like to think of as generation 1, where we've used INKmune to activate NK cells outside the patient's body, and then given those activated NK cells to the patient. And in both of these trials, left-hand one being in the UK and London, and the right-hand one being a multi-center trial across the United States, both of them in acute myeloid leukemia. And what we've shown here, and there's a lot of data, but I just want you to focus on 2 elements of it. The first paper on the left-hand side in PLOS One, we can see that in the bottom plot, the spontaneous lysis of resistant Raji cells, here we've looked at 4 patients post-infusion of donor cells. Now, these NK cells that were activated with INKmune outside the body actually came from related donors. They were not the patient's own NK cells. And so we could trace them in the patient, and we demonstrated that they didn't hang around for very long. Like most, or like every study done with adoptive transfer of NK cells, they actually survive in the peripheral blood for a very short period of time. So the fact that at 8 weeks, in this case, patient 01, patient 03, and patient 011, 8 weeks after the infusion of the donor cells, a substantial number of the patient's own peripheral blood NK cells are activated. And not are they just only are they activated, but they can kill the NK-resistant cell line Raji. So they're functionally activated, even though the donor NK cells disappeared from their circulation a very long time ago. And the plot next to it shows the duration of activated NK cells in the peripheral blood of those patients, and the fact that they have very high levels of the NK cells in the peripheral blood are expressing CD69 and activated, even though there are no donor NK cells left at that stage. And we have similar data from the US trial, and I'll just point you to the example in the middle there. This is a patient who at day 56 had only 0.0187% of the NK cells in his blood were from the donor. And yet, as you can see directly beneath that at day 56, around about 12% of the total NK cells in his blood were activated. And so these are the patient's own NK cells that are activated even in the absence of donor NK cells. And we believe this is a critical mechanism of action of INKmune. We've gone back to the laboratory to look at how this might happen, and what you can see here are the evidence of that. So when we incubate resting NK cells with resting NK cells, they do come together over 4 hours, and they bind to each other and they form conjugates. And here you can see about 70 NK cells formed conjugates with other NK cells. But if we take a tumor-primed NK cell and incubate it with a resting NK cell over the same time period, about 120 of them bind. And here you can see a single snapshot from that 4-hour incubation. Here, the resting NK plus resting NK, and there are a few conjugates. Here, 3 of them in that snapshot. And here in a random snapshot from resting NK plus tumor-primed NK, you can see how many more of them are interacting. And what that leads to, as you can see here, is the CD16 shedding is associated with NK cell activation. And here, the proportion of NK cells expressing CD16 goes down far greater when activated by binding to a tumor-primed NK cell. So this NK cell carrying the INB16 membrane signals is offering those signals up to resting NK cells and activating them, as you can see here. And here, over a much longer period, where we sustain the NK cells with low-dose IL-15, we get a little bit of activation with IL-15, but in the presence of tumor-primed NK cells, the proportion of NK cells that are activated goes up to over 50%. We now have one of the mechanisms of action of INKmune in vivo. We've looked at the genome, the genes that are upregulated when NK cells bind to INKmune. This paper was published in 2017, and what you can see here are the comparison of NK cells activated with K562, the conventional or the prototypical NK cell target cell, activated with CTV1 or INKmune, or activated with IL-2. You can see there are 183 distinct genes which are switched on by CTV1 stimulation that are unique to CTV1 or INKmune priming, and 164 that are downregulated. And what the outcome of all of this is actually a degree of cytokine secretion. So we see an increase in gamma interferon after CTV1 priming, which we don't see with K562. We see an increase in TNF alpha, MIP1 alpha, MIP1 beta, RANTES, etc. And we'll come back to these later in terms of what this might mean in the patient. Those genomic changes, those changes in mRNA transcription, are also associated with changes in proteins. And here we've looked at resting NK cells versus those that have been activated by co-culture with INB16. And you can see, as you would expect, upregulation of the CD25 molecule. This is the IL-2 receptor, and it makes them more sensitive to IL-2 stimulation. Upregulation of CD69, the activation antigen. Interestingly, and actually quite intriguingly, downregulation of NKG2D. Now, this is a ligand for a lot of tumor associated molecules such as MICA and MICB, and we would expect cells that have got a downregulation of NKG2D to be less functional, and yet our cells are more functional. And the answer to that conundrum is that when INKmune binds to the NK cell, it actually ligates the NKG2D receptor, and it sends that signal already to the NK cell. This means the NK cell can bind to a target cell, and it's already received some of its signals through NKG2D, and we believe that's part of the mechanism of action of the enhanced killing. Similarly, the other NK receptors like NKp30 are also downregulated. There's a substantial upregulation of ICAM1, and this is really important because this is a very important adhesion molecule for NK cells when binding to their target cell. And I think you'll see later on that this is demonstrated in both visually and numerically in terms of the avidity, the strength of binding of the NK cell to the tumor cell. What we also see is shedding of CD16, and that's well known as NK cell activation through natural cytotoxicity receptors is associated with shedding of CD16, and there's a good rationale for that, and upregulation of the NK differentiation marker, maturation marker, CD57. So these look like NK cells which are ready to lyse a tumor cell. Of course, the other aspect of the tumor microenvironment that these NK cells are going to end up going into in the solid tumor setting is hypoxia. We know that the oxygen concentration within a tumor is substantially lower than the oxygen concentration in the peripheral blood, and so these NK cells have to be able to cope with hypoxia. And what we've looked at here is, again, the ovarian cancer cell line of CAR3, which we know is highly resistant to NK cell killing. And here the NK cells have come from the ascites of ladies with ovarian cancer. Now, the ascites is a site of the tumor in these women, and it is hypoxic. So these NK cells are in a hypoxic microenvironment. If we take them out of that microenvironment, put them into normoxia, into a conventional killing assay in a laboratory, and test the resting NK cells for killing of the OVCAR cell line, you can see that at 6 hours, 16 hours, 24 hours, and 48 hours, there's very little killing by resting NK cells. If we prime those resting NK cells in hypoxia with INKmune, we can substantially increase that killing at all time points. But very interestingly, if we do the whole experiment in hypoxia, so we take the NK cells out of the hypoxic microenvironment and we put them into a killing assay in hypoxia, these NK cells, even at rest, are substantially better than the same NK cells running that assay in normoxia. In other words, the NK cells are conditioned to hypoxia, and they perform better in hypoxia than they do in normoxia. But intriguingly, we can make that even better by priming them in hypoxia with INKmune. And so this leads us to believe that INKmune priming will maintain an NK cell in hypoxia even better than an NK cell that's conditioned to hypoxia in the first place. Now, INKmune isn't the only reagent that does this. IL-15 also enhances NK cell function in hypoxia. But what we see here is that INKmune is substantially better at increasing killing than IL-15 is. The data you've seen so far have largely been in ovarian cancer in terms of the solid tumor, but also a small bit of data in ovarian cancer and in breast cancer. But here we show that in renal cell cancer, this is a renal cell cancer 7860 and another one ACHN, where NK cells really, resting NK cells, these are from healthy donors, really struggle to provide any killing at all. And yet tumor priming with INKmune substantially increases that in all cases. And the same is true in nasopharyngeal cancer. And we're lucky here to have 2 nasopharyngeal cancers from colleagues at the Chinese University of Hong Kong, where you see exactly the same phenomenon. Tumor priming overcomes the inhibition from that cell. So going back to which genes are upregulated when an NK cell is activated by INKmune, you can see here that there are 163 genes which are uniquely upregulated and 209 which are downregulated. If we look first at the upregulated genes, what excited us was that came out very strongly was upregulation of CXCL10, CD70, and STAT5B. And here you can see publications from other groups explaining why that's significant. CXCL10 activation of NK cells allows them to kill dormant tumor cells that resist T-cell lysis, and STAT5 is essential for NK proliferation and killing. Whereas CD70 is the ligand for CD27 and directly induces natural killer cell activity. So these are good, provide good rationale for why INKmune primed NK cells should be so good. And in addition, IL-2 downregulates CXCR4, CXCR2, and BCL6, proteins which are associated with movement of the NK cell from the peripheral blood into extravascular sites where tumors are, and survival proteins like BCL6. We've gone on now to look at the next obvious target, which is the proteome. So changes in the genome, changes in the mRNA should lead to transcription and changes in proteomics. And here we've used conventional proteomics analysis with a company called Biosignosis to look at what happens when you activate cells either with IL-15 or with INKmune, INB16 here. And looking at proteins associated with NK cell lytic function, target cell killing, we can see resting NK cells, as you might imagine, no upregulation of these proteins. But here, IL-15 treatment upregulates many of the proteins associated with killing, downregulates some. INKmune upregulates the same proteins. So it's not surprising that INKmune primed NK cells have enhanced killing, just like IL-15 activated NK cells. But what was really exciting and indeed surprising to us, since we hadn't looked for this, it just came out of the data analysis. IL-15 barely upregulates proteins associated with mitochondrial survival, whereas INKmune or INB16 substantially upregulates nearly all of the proteins associated with mitochondrial survival. Now, the mitochondria are the engines of the cell. They're the organelles which take oxygen and drive cell survival. What we know from other publications is that dysfunctional NK cells in the tumor microenvironment are known to have adverse or impaired mitochondrial function. So it's a hypothesis that improvement in mitochondrial survival proteins will overcome that intratumoral impairment of mitochondrial function, and we expect to have NK cells that perform better in the tumor microenvironment. So we've taken this further and we've compared resting NK cells with the same IL-15 activated NK cells and NK cells activated by IL-12, IL-15, and IL-18, the so-called cytokine-induced memory-like NK cells. And we know these cells are very similar to our tumor-induced memory-like NK cells that we get after INKmune priming. But just looking head-to-head, our INKmune prime memory NK cells versus cytokine prime memory NK cells, there are some substantial differences which could be very informative and certainly support our hypothesis that our cells will do well in the tumor microenvironment. So this is looking at the relative increase in these proteins compared to the same protein in cytokine-induced memory-like NK cells. And you can see that proteins such as CeRu inhibit IL-17 secretion, which is an immunosuppressive cytokine, and interferon gamma, as everyone knows, upregulates interleukin-12, interleukin-23, and TNF, all important in NK cell survival, proliferation, and function. There are other proteins associated with NK cell activation and cell-cell signaling. This may speak to the NK interactions that we see enhanced. Increased regulation of increased cell proliferation, regulation of gamma interferon production, which then feeds back to the gamma interferon story here, increased IL-2 production, and increased leukocyte migration and extravasation, and then more cell proliferation and inhibition of the inhibitory cytokine IL-10. So there are 9,600 proteins involved in this proteomic analysis, and we're working through them at present. But already we have evidence of some proteins which suggest there is an improved performance of these NK cells in the tumor microenvironment. In fact, these data support that probably even more. This is looking at the metabolism of the NK cell and the ability of those mitochondria to respond to stimuli. And these are compared to the background, the unstimulated. So this is relative increase or decrease compared to the background. And here you can see that both tumor-induced NK cells and cytokine-induced memory-like NK cells have increased mitochondrial respiration compared to resting NK cells, as you would imagine. The maximal respiration is slightly higher in tumor-induced memory-like NK cells, but I think the most important data here, the most informative, are that NK cells activated by INKmune have increased spare respiratory capacity compared to NK cells that are activated by a panoply of cytokines. And this, we believe, is partly due to that increased mitochondrial survival protein associated with INKmune priming. It may also be due to nutrient receptors. So we know that cytokines don't induce nutrient receptors to the same degree that INKmune does. And here we can see 37 nutrient receptors upregulated within 16 hours of binding to INKmune compared to 1 after cytokine stimulation. That speaks even more to the tumor microenvironment story here, where this protein LRP1, which enhances mitophagy, this is the internalization, the digestion of mitochondria in impaired NK cells. This is upregulated by IL-15. It's a clearance mechanism for dying mitochondria, whereas INKmune doesn't do that. In fact, INKmune upregulates BNIT1, which is a promoter of mitochondrial fitness. And that protein is not upregulated by IL-15. This is one of the mitochondrial survival proteins that I described just now. So more evidence that this arming of the NK cell by interaction with INB16 and the transfer of the proteins from the INB16 membrane is involved in priming an NK cell, which should have better performance in the tumor microenvironment. So what we've been interested in here is the ability of the NK cell to bind strongly to the target cell. Plainly, for an NK cell to kill a tumor cell, it has to find it, it has to bind to it, and it has to stay sufficient bounds for a sufficiently long time to have an effect and to deliver its killing function. So here we bind the NK cells to the target cell, and then having bound them, we apply a force across that co-culture and measure the NK cells as they come off that binding. And so here in black, you can see very little force, something like 40 piconewtons is enough to disrupt 98%, 95% of the NK cells that are bound to SCOV3, the NK resistant target. And if we add IL-2 to that, we incubate the cells with IL-2 and activate them, they bind a little more strongly. But if we incubate them with IL-15 or with INKmune, we increase that substantially more. And taking this graph here and turning it into solid figures, you can see that resting NK cells bind very poorly. And indeed, if you look at their ability to kill against this axis here, we have very little lysis. If we activate them with IL-2, we get slightly stronger binding, but virtually no increase in killing. But if we incubate them with INB16, you get stronger binding and greater lysis, and IL-15 also causes stronger binding and greater lysis. And so the lytic function is a direct correlate of the degree or the strength of that binding. If we take that and try and visualize it, you can see here NK cells from a patient with ovarian cancer being incubated with SCOV3 tumor cells in culture, the NK cells being the blue cells. And here you can see the NK cells interacting with the tumor cells, rolling along them, but never forming a strong synapse, never forming a strong, highly avid bond, and therefore not killing the tumor cells. In contrast, slightly higher magnification, if we incubate those cells beforehand with INKmune overnight and then put them into the same culture, what you can see here is an extremely avid bond. This tumor cell is doing its utmost to get away from the NK cell. The NK cell refuses to break the bond, and as you can see within time, here it goes, the tumor cell starts to break apart and die, and there we have a dead tumor cell. If we take that into a long-term assay, here we've taken those same SCOV3 tumor cells, we've grown them for 24 hours until they coat the plate, we've then added NK cells, and to half the wells, 12 hours later, we've added INKmune. And so the wells that have not had INKmune added are in blue, and you can see in terms of cell death, even as a high ratio of 20 NK cells to one tumor cell, we get no tumor cell killing. However, in contrast, if we prime them with INKmune during that culture phase, and these tumor cells are still growing in the presence of the NK cells plus INKmune, here you can see this is the degree of lysis of those cells, and even at 5 NK cells to one tumor cell compared to 20 here, we have very, very substantial killing. So we've heard a lot in recent years about multiple cytokines being used to provide a memory-like NK cell population, and I think most of those data have come from Rizwan Momri and Todd Feniger's group. And this paper by Rizwan from 2016 defines the phenotype of a memory-like NK cell using IL-12, IL-15, and IL-18. And you can see there's an increase in expression of CD56, a shedding of CD16, as we've said before. This is associated with NK cell activation and upregulation of these markers. And what we find when we prime with INKmune, we get the same phenotypic. We get downregulation of CD16, upregulation of CD94, upregulation of NKG2A, as you might imagine. And very intriguingly, this increased CD56 population that one sees, we see them here. So this is CD56 expression. These are regular CD56 expression on NK cells, and these are the hyper CD56 cells that we generate after overnight stimulation with INKmune. We find the same phenomena in vivo. So these are data from the first patient treated in the current UK trial of INKmune, where we demonstrate that we obtain memory-like NK cells in vivo. And to remind those of you that have seen these data before, this is the first patient was a patient with myelodysplastic syndrome who was entered into the trial in April 2021 and treated in June that year. But what you can see here in terms of his history, he presented with disease in March 2018. He was treated with high dose with azacitidine through 3 conventional treatments, a total of 19 doses over that period of time. And during that time, his circulating tumor DNA increased and the number of blasts in his peripheral blood increased, such that when he joined our trial in 2021, 17% of his bone marrow had 17% blasts. His ECOG status was 2. This is a readout of clinical fitness. ECOG 0 being healthy, being normal, ECOG 5 being moribund and close to death, and he was an ECOG 2. What we did was on the 7th of July, he received his first INKmune infusion, and then the second one 7 days later, and the third one 7 days later, after which he had no more INKmune infusions. They were all well tolerated, as in fact, the most recent patient has been treated as an outpatient. What we saw after treatment was a mild reduction in his circulating tumor DNA, a mild reduction in his blast count, but most importantly for him was a resolution of his ECOG status to ECOG 0, with reduced transfusion independence, reduced transfusion requirements rather, and fully active, back to playing badminton with his friends. And this is a 78-year-old man who was previously fit prior to developing MDS. So from very, very significant improvement in quality of life, which he attributes to the drug. What we saw were really interesting biomarker data. So here we're looking at the NKG2D positive NK cells, those which target known tumor antigens. And we can see at screen that very few of these NK cells were activated, and within, by day 29, day 29 here rather, you can see over 60% of his NKG2D positive NK cells were activated before falling and then recovering. Now, bearing in mind his last treatment was day 15, the fact that they fell and recovered suggests that there is an ongoing effect of INB16, of INKmune in this patient. There is no INKmune in this patient by day 43. It can only be molecules that are carried on the surface of circulating NK cells, which have trogocytosed the INKmune, and you can see we get activation of CD16 positive NK cells as well. Very importantly, when we take those NK cells out of the peripheral blood of the patient, prior to day 15, we got no NK cell activity. That by day 15, we started to see NK cell function, and really importantly, these NK cells could not only kill NK sensitive targets, but had the ability to kill NK resistant targets, which gives them that magic phenotype of being a memory-like NK cell. But what's very interesting and very satisfying from my perspective is the cytokine profile associated with these findings. These are systemic cytokine levels in the patient. So these are samples taken from their blood, and you can see MIP1 alpha, MIP1 beta, TNF alpha, all increasing up to that day 29 and then falling after day 29. And suggesting that this is a real phenomenon here that we saw with the activation as well. And I think this is due to the secretion of IL-2 receptor alpha, which is associated with NK cell function. The secretion of IL-2 receptor alpha makes the NK cells, it's a sort of positive feedback loop. And the recovery after day 29 is almost certainly due to the resolution of the IL-2 receptor alpha levels. And if we go back to those original data I showed you earlier on today, what we see here is that the same cytokines that we saw in the laboratory back in 2007, that INKmune induces MIP1 alpha, MIP1 beta, TNF alpha, IL-2 receptor alpha, we see exactly the same upregulated in the patient after treatment with INKmune, which is extremely satisfying. So in conclusion, NK interaction with some tumor cells, such as CTV1 or MB16 or INKmune, provides the necessary priming signals to drive them to a memory-like phenotype, which we've called tumor-primed NK cells. And these cells target hematological conditions, we know that, but also solid tumors in vitro. And they can be generated not just from healthy donors, but as you've seen here, from patient NK cells, which is obviously the mechanism of action that we're seeking with INKmune trials. They're not inhibited by self-MHC once they've been primed, and that's also true of cytokine-induced memory-like NK cells. They do show increased activity even in extreme hypoxia, and those assays I showed you were in 0.1% oxygen. And although I didn't show you the data, they're not inhibited by MDSC or Treg cells, which we know are in the tumor microenvironment and have the capacity to inhibit NK function. The proteomic analysis is very, very interesting and still being investigated by us, but the mitochondrial survival protein upregulation, the nutrient receptor upregulation, the upregulation of lytic pathway associated proteins and proliferation associated proteins, all support what we've seen both in vitro and in vivo, and we will be developing IP around those, I suspect, in the near future. But this enhanced metabolic profile that comes from these changes is going to be critical in the tumor microenvironment, and is comparable in terms of function to cytokine-induced memory-like NK cells, but has this increased spare capacity, which I think, as I said, goes back to the upregulation of mitochondrial survival proteins that we've seen. The 4 patients we've treated so far have received INKmune, very well tolerated. It's extremely safe. It certainly appears to induce memory-like NK cells in vivo, in that they gain the function to kill NK-resistant tumor cells. And we have this systemic cytokine secretion, which matches the in vitro data, which is extremely satisfying and is associated with the long-term persistence of these activated NK cells in 3 of the 4 patients we've treated so far. So with that, I will end the webinar. I hope you found it as interesting as we found gathering the data and presenting them to you. And we look forward to any questions you want to submit to the members of the team. Thank you very much indeed.